But I know people are designing the trials now from a pharmaceutical way.
Thank you. Okay, thank you very much.
Thank you.
Now, it's my pleasure to introduce you Juan Rodriguez. Most of you know Juan Rodriguez, but I would like to tell that he is the leading author and the leading scientist of the group who first published that there was bacteria in human milk in 2003. That human milk was the source of lactic acid bacteria. And now we will see what happened in 16 years. So please, Juan.
Thank you very much, Hertag. So in this year, 88, when we started to work in human microbiota, we thought that there was a microbiota only in a few human locations. In the mouth, in the nose, in the vagina, and most in the gut. In fact, in a popular magazine in Spain in this year, not talking about the benefits of human milk, they say that it's a sterile food. No, and it's surprising to talk about the benefits of human milk. They have here a photograph of a breastfeed baby. So now. 30 years later, the picture has changed a lot. Now we know that any mucosal surface that produces a biological fluid in our body has a microbiota. Of course, there are some places, like the placenta and this kind of locations, that are the subject of a very strong controversy at present. But anyway, we also know that these ecosystems are submitted to exchanges. The exchanges between microbiotas are common in the human being. And of course, now it's widely accepted that human milk has a microbiota, that human milk is one of the providers of the first microbes that a baby receives in life. Of course, there are many places, many labs around the world working at present in this issue, in the human milk microbiota. As far as know at present, with the data that we have at present, we know that the human milk acquires microbes in the last third of pregnancy because of different adaptations that I will talk later. And this microbiota remains stable as long as milk is produced. Then when weaning starts, this population declines, and after complete weaning, these microbes completely disappear in the breast. So this makes the human milk microbiota a very peculiar microbiota, because it's the only human microbiota that we know that is specific for a certain period of life, for late pregnancy and lactation. And the second property of the human milk microbiota is that the concentration is quite low, especially if we compare with that in the vagina or with that in the gut. Usually there is around 10 to 3 colony-forming units per milliliter of milk, and this means that this concentration causes no harm in the breast of a woman. When we culture milk samples, human milk samples, up to the present, we have been able to isolate more than 50 different bacterial genera, more than 200 different bacterial species, and including new species like Streptococcus lactarius or some others that will be published just quite soon. So by cultures, we can see that the dominant bacteria in human milk are Streptococcus, Staphylococcus, related gram-positive bacteria, but also some lactic acid bacteria and bifidobacteria. This first group of bacteria are common in milk worldwide, while lactic acid bacteria and bifidobacteria are common in developing countries, but less common in developed countries, and it's thought because of the antibiotic exposure and other factors. Anyway, all these microbes are considered the core microbiota of human milk. Then, when we check for DNA, for nucleic acids, then we also usually find in human milk DNA from the microbes that I told before, for those that we can culture in the lab. But in addition, we also find DNA from bacteria that are closely associated to the gut. Fecalibacterium, Roseburia, Eubacterium, and this kind of things. And of course, we cannot find them in cultures because at present we are not able to culture them. It's very, very difficult to culture them in the lab. And in addition, by DNA methods, we also find a third group of bacteria that is usually related to the soil and to water. This kind of bacteria, we can easily culture them in the lab, but they are not present in the milk when we culture mixed samples. Anyway, we detect the DNA. Nowadays, it's generally considered that this kind of bacteria probably are a technical artifact. Why? Just because DNA from soil and water bacteria are common in the molecular biology regions and in molecular grade water. So when we are studying microbiotas with a low biomass, such as human milk, this, of course, as I told you, can be just a technical artifact. And this makes very important to introduce right controls when in the studies of the human milk microbiome. Anyway, I like to highlight that the human milk microbiome is not the result of just a random contamination. When we study a human milk microbiota, we found that it has a well-defined structure with well-defined consortia. So this is also something important. Of course, most of the times when we talk about microbiome, we are actually, at the end of the day, working with bacteria. And there is DNA in the milk from other kind of microbes, from viruses, archaeas, molds and yeast, and also from protozoa. And of course, when we talk about bacteria, we found 5 main phyla in human milk: Bacteroidetes, Firmicutes, Actinobacteria, Proteobacteria, and Verrucomicrobia. However, please have in mind that most of the times in microbiome studies, we talk about phyla or phylums. But the taxonomical level that provides the highest flexibility in a microbiome is the strain level. The strains are very important. And so just as an example in our lab, for example, we can find in human milk one phylum, this Firmicutes, and we can isolate bacteria from the genus Staphylococcus. And of course, we found in the same sample from a woman, Staphylococcus epidermidis and Staphylococcus hominis. And then after genotyping, we found that the same sample contains 12 different strains of Staphylococcus epidermidis and 4 different strains of Staphylococcus hominis. And please just have in mind that each strain may have different properties. Anyway, I'm going to talk just for a few slides about the origin of the human milk bacteria. Of course, human breastfeeding means intense physical contact between the mother and the infant. So, when we talk about bacterial milk, we usually think in 2 main sources: the oral mouth or the skin of the mother or the baby. So, in relation to the infant mouth and also the nasopharynx, we also think about streptococci because it's very rich. There are many streptococci in these places, particularly of 2 groups, the Mitis and Salivarius streptococci, and related bacteria. However, streptococci don't live only in the mouth. For example, they are very abundant in the gut. So, some data in our lab show that we don't know if streptococci go from the infant mouth to the milk or if it's through the opposite way. if the streptococcus in the milk can go first to the baby's mouth. So very recently, a few months ago, we published this study in which we study samples of precalostrum, of course the calostrum produced before delivery in first pregnancies. And we study by whole genome sequencing of those strains belonging to a species that we found both in the mother and the child. And the results of the study is that in most of these mother-infant pairs, Streptococcus and Staphylococci as well, strains could be isolated first in precalostrum and later in the baby's mouth. So we know that this way is happening, and the opposite way we don't know because there has been no study so far focused on this subject. The second instinctive source of microbes for the milk is the skin of the mother and also of the baby, as a source mainly of Staphylococci, particularly coagulase-negative Staphylococci, and mainly Staphylococcus epidermidis, and also Propionibacterium, Corynebacterium, and related bacteria. So, but also, again, have in mind that the place in our body when we have the highest concentration of Staphylococci are not our skin, is our gut. Anyway, as Sertak told before, in the year 2003, we found that in the milk there were bacteria that we were not expecting in this biological fluid. These are lactic acid bacteria. And then just after realizing about this, we started a study with a hospital in Madrid involving both vaginal and C-section delivered babies and checking any kind of sample, perinatal sample, just to study the source of the bacteria for human milk. And then this study was limited to lactic acid bacteria and include genotyping techniques just to see if there is a transmission to the strain level. And we found that vaginal exudates were different to those found in the breast skin and also different to those found in the milk. However, we found lactic acid bacteria in maternal feces that were also found in the milk and were also found in the infant of this feces. Later, we also found not only lactic acid bacteria, but some bifidobacterial strains that are strict anaerobes and that they don't live in the skin or don't live in the infant mouth. So nowadays, it's considered that bacterial communities that exist in human milk skin and, of course, in the mouth and in the nose can share some phenotypes, but also that there are many differences between these different places within the same mother-infant pair. So the rest of the bacteria, where they come from? But I guess that the answer is in the maternal gut. This paper in the year 2001, when we were thinking how this bacteria can reach the mammary gland, show that dendritic cells in the lamina propria of the gut can send dendrites and can trap some bacteria in the lumen of the gut and then go again to the lamina propria and spread to other locations. This study involved at that time non-pathogenic bacteria in healthy hosts. So when we read this paper, we send a person of our group to the group that developed this technique to see if this was possible during pregnancy or lactation. And we found that, of course, some bacteria found in human milk, when given orally to a pregnant woman, can attach to the dendritic cells, but also to macrophages, and in this way, they can go across the mesenteric lymph node and appear in human milk. Therefore, that there is a physiological bacterial translocation from the gut to the mesenteric lymph nodes and later to mammary gland during pregnancy and lactation. And the same study has been repeated 4 times in different labs across Europe and the States, and the results have been very, very similar, as you can see, for example, in this photograph, how the bacteria can go across the blood and arrive to the mammary gland attached to this kind of cells. Of course, from fertilization to delivery, as we all know, there is a lot of changes, wonderful changes that happens, but just parallel, there are different changes in the body of the woman, and these adaptations affect to all organs, all tissues, all systems, but of course affect the whole digestive system, from mouth to the gut, to the cardiovascular system, to the immune system, of course to the uterus, and of course to the mammary glands. And all these adaptations, without any exception, predispose to a physiological translocation of bacteria that we know that it happens also in healthy hosts. Anyway, we know that this phenomenon is increased in this period of life. And it doesn't mean, like happens with pathogens that are very aggressive, and then there is a disruption of the mucosal surface of the gut. It means a very elegant mechanism that involves no destruction of the gut mucosal surface and involves a very complex interaction between the epithelium of the gut, between immune cells, and between certain bacteria. Because it's a selective process, it means that only a few bacterial groups can reach this mechanism. Of course, I've been talking about bacteria from the mother, vertical transfer of bacteria from the mother to the baby. But of course, we know that there are microbes in milk that cannot come from the mother. Of course, if you think, for example, if a mother is pumping the milk, most of the times at homes, the pumps are rinsed just with drinkable water, and drinkable water has microbes, and at concentration much higher than there are normally in human milk, and usually involve gram-negative bacteria, yeast, that can reach concentrations quite high, even 10 to 5, 10 to 6 colony-forming units per milliliter. From the water, and of course, if the practice of collection milk are not hygienic, also is a source of bacteria for this biological fluid. We also know that there are many factors that may modulate the milk microbiota, and probably there are many others that we don't know but play a role. Host factors, microbial factors, environmental factors, socioeconomic factors, medical factors, and anything you can think about this, anything you may have in mind. So, when we always want to define something as normal, and it's very difficult in this issue, of course, define a normal human milk microbiota, because women with different milk microbiome profiles may have a normal human milk microbiome. So, if the composition does not serve as to define a healthy human milk microbiome, how we can define a healthy human milk microbiome? And in this way, it's similar to what happened with the gut microbiome. Well, we can define a healthy human milk microbiome because of the roles that we expect for the human milk microbiome, the roles for the baby, but also the roles for breast health. In fact, when we study different microbes isolated from human milk, we see a lot of different functions, from protection against infection, neuromodulation, immunomodulation, production of substances that has benefit to the baby, but of course, all these roles are strain-specific. So they are not common for any bacteria that we isolate from human milk. It depends on the strain. And nowadays, we are carrying an European project just to try to detect the microbial functions that are common in human milk worldwide. So the minimal function that we can see in any milk from any mother. Of course, as is normal, human milk bacteria are transferred to the baby and may play key roles once inside the infant gut. This paper was quite relevant because it showed that the cessation of breastfeeding drives the maturation of the infant gut microbiome. before this study, we thought that the introduction of solid foods was what changed the human gut microbiota, although the woman was still breastfeeding the baby. But this study showed just the contrary, that the microbiota of babies is dominated by milk bacteria as long as the baby is breastfed, independently that we have introduced solid foods. So the impact of the human milk bacteria on infant gut may be stronger than we previously thought. I also like to say that sometimes it's very difficult to separate the roles of the different milk compounds. I mean, how to study only milk bacteria and not other milk compounds that are with the milk and are actually, at the end of the day, is what the baby is taking. So I mean that, for example, when we talk about antimicrobial activity may be played by different substances and also from bacteria in human milk and also in a synergistic way. So I mean the complexity of human milk and how many roles are shared by different elements. Of course, as happened in the gut microbiome or in the vaginal microbiome, human milk can also have a process of dysbiosis, an alteration of this microbiome. And this happens when there is mastitis, when there is painful breastfeeding, this kind of conditions. So normally, what we find in a human milk sample is a high diversity, but at a low concentration, 10 to 3, 10 to 2 colony forming unit per milliliter. So when we found a problem, what we usually find is that the diversity just decreases sharply. There is now a low diversity, but at a high concentration. It means that maybe 1, 2 strains of Staphylococcus, Streptococcus can increase a lot, reaching concentration of up to 10 to 6 colony forming units per milliliter. It's quite difficult to reach higher concentrations because at 10 to 6 per milliliter, mammary gut is just full of bacteria. There is no room for more bacteria inside. And we can see this just by classical techniques, for example, optical microscopy. You can see here a sample of milk from a mother woman with mastitis, a lot of Staphylococcus, and a lot, of course, of T cells and also epithelial cells, because there is an inflammation going on. And then we can compare with a sample of milk or from a healthy woman. You can see bacteria, but the concentration is much lower, and also we can see immune cells, but also the concentration is lower. Of course, we also can see this effect, the loss of diversity, with culture-independent techniques. This is the study of the microbiome of a woman with a recurrent mastitis, and you can see here that when she is with mastitis, there is no diversity. Most of the sequences are from the staph that cause the mastitis. When she is recovering, the diversity increase is maximum when she is feeling well, but again, is almost lost when in the following episode. Of course, we can see also this effect by electron microscopy. Here you can see the biopsy of a healthy woman is a mammary duct, and you can see here that there are bacteria, but just a low density, and some of them are interacting with the fat globules in the milk. But for example, in a woman with mastitis, what we see is a very, very complex and very dense biofilm from inside the mammary duct. Just the last 2 slides is to talk a little bit about preterm babies. Of course, this is just in the documentation that in the neonatal unit of San Diego in the USA provide to women, mothers of preterm babies. And in this information, this is the things that human milk may have that infant formula does not have, don't have, that can be relevant for infant health. And you know, the last piece that they added is because human milk contains bacteria. You know, maybe 15 years ago, if we know that milk contains bacteria, maybe was a reason for not breastfeed. And now it's a reason to give milk to even to very low preterm babies. So the administration of human milk microbiota to preterm babies, just to finish the last slide, can be done in different ways. The optimal way is providing the own mother's milk. Of course, has the own microbiome of this mother, and is a quite personalized way of transfer of these microbes. And of course, the second one is the milk of a donor, but milk that is not being pasteurized. Of course, this is not common at present in the human milk banks worldwide, but it's a trend. There is an increasing number of banks that, for example, they are using not pasteurized milk for very, very selected donors after a screening, of course, of cytomegalovirus by PCR, both in blood and in milk. But as I told you, in most cases, the milk is submitted to a pasteurization, and of course, this heat treatment destroyed the human milk microbiota. So in these cases, what to do? Well, there are different options. The easiest at this stage nowadays is the use of human milk-derived probiotics, that there are just a very few at present. But also, I hope that in the next years, we can use human milk minimal microbiotas, or the use of what is called a milk microbial transplant. So there are strategies that can be used to give this bacteria to babies that otherwise would not have contact with them. So of course we can add as a supplement to the milk, or alternatively we can give them directly to the babies. So just as a conclusion, there is human milk contains a microbiota, a site-specific microbiota, with a core microbiome comprising a few abundant genera, but also with a high individual variability. Also, this microbiota can suffer dysbiosis processes, but it can be modulated. Of course, both external and internal routes can explain the presence of bacteria in human milk, and as far as we know as present, probably this microbiota may play relevant roles, although they are quite unknown at present. So the future means a lot of research, a lot of clinical studies. I mean that our knowledge of the human milk microbiota is just starting at present. So there is a lot of work to